In this video, we'll look at setting up your data for analysis with the SCR package. SCR is an R package for fitting spatial capture-recapture models. It's a very detailed package that can fit a lot of different kinds of models, and what we're going to look at in this video is just setting up data for the simplest kind of model. There are three main input files that you need to enter. One is a file that contains the locations of camera traps and any covariates associated with those traps. The second file contains the capture histories, that's which animal is detected at which trap. And the third file is optional, and it contains your habitat mask. The camera trap input file contains information about the cameras. Each row in the camera trap input file contains the location of a camera, plus any extra information about that camera. So a typical camera trap input file for 3 cameras might look like this. The first 3 columns in the camera trap input file are compulsory, and they contain the trap ID, that's the unique identifier for the camera, and the X and Y coordinates of each of the cameras. Those 3 variables have to appear, and they have to appear in that order. We can also include detector covariates in the camera trap input file. These are covariates that affect detectability at each of the cameras. Detector covariates are indicated to the right of this / sign. So here we have 2 detector covariates, the terrain roughness index and temperature, and we'd include these if we think that they have an effect on the detectability of animals at each of these cameras. Camera trap input file can be saved in a variety of formats. The easiest way is just to save it as a text file with spaces separating the values and with a header row starting with a hash. But you can also save it as a CSV file and as an Excel spreadsheet. So just to give you an idea of what the text file format would look like, for this case it would look something like this. The capture history input file contains our detections. Each row contains a record of a particular detection, and it basically just records which animal was seen on which camera. So let's have a look through an example input file, column by column. The first column contains a session identifier. Even if you've only got one session, this column must still be included. The second column is an animal identifier. This identifies which animal was recorded for a particular detection. The next column is an occasion identifier, and again, you have to include this column even if you only use one occasion. The fourth column is a trap identifier, so it's an ID for which camera the animal was sighted at. There are 2 different ways that you can record the ID of a trap. Either you can use a categorical ID that relates back to the ID that was used in the camera trap input file, or you can record the XY coordinates of the camera. If you're going to use the XY coordinates of the camera, you'd replace this trap ID column with 2 columns, one for the X and one for the Y coordinates. Again, you can save this as a text file or as a CSV or Excel spreadsheet. So in the text file format, a capture history input file would look something like this. The third input file that we need is the habitat mask. Remember, the habitat mask is just a set of square grid cells that represent habitat in the vicinity of the detectors. The reason why we need a habitat mask is because spatial capture-recapture models involve maximizing a likelihood, and this involves integrating over space. The way that a computer integrates over space is to take that space and to chop it up into grid cells and then sum over those cells. The most important property of a habitat mask is that animals that have activity centers outside the boundary of the habitat mask should have a very low probability of being detected at any of the cameras. If we zoom into the habitat mask, we can clearly see this grid structure. And the way we're going to represent each of these grid cells in the mask is by the center of each of the grid cells. So a common question is, how big should the mask grid cells be? And this is really a trade-off. If we want to approximate the integrals that SCR use as well, the grid cells should be small, but this increases the time it takes to run models. And so a rough rule of thumb is to use a mask spacing somewhere between a half and 1 sigma, or to try for somewhere between 1,000 and 3,000 grid points. Sometimes it won't be possible to fulfill both of these goals, and that will depend on the size of the study area, but these are just rough rules of thumb. So in terms of what a mask input file should look like, The first two columns of the mask input file contain the X and Y coordinates of the center of each of the mask cells. The remaining columns capture mask covariates, and these are usually used to model density. Again, the mask file can be saved as a text file, CSV, or Excel spreadsheet. If we use the text format, this is what it would look like. Once we've got our 3 input files, all that we need to do is to read these into SCR. And we can do this with 2 functions, read.captest and read.mask. Read.captest reads in both the capture history file and the camera trap input file. There are a bunch of other options whose values will depend on your particular application. For read.mask, all we need to enter is the name of the mask file and the spacing between mask cells. You can leave out the spacing and SCR will try and work out the spacing, but if you know it, it's safer to add it. So let's run through these 2 lines of code for this particular example in the R console. Once we've loaded the SCR library, we can just run read.captest, and if everything works fine, you'll get this message that no errors have been found. We can also run read.mask. If we want to check that everything's worked okay, we can plot the detectors. Here these plots won't make much sense because we've just got 3 camera traps, but it will plot the detectors. We can plot the capture history and confirm that we've got 2 occasions, 3 detections, and 3 unique animals. We can also plot the mask. If we zoom in on the mask, we can see that it consists of 4 grid cells, as we know that we've got in our text file. Once we've loaded in our input data files, we can proceed on to analysis, and this will be covered in other videos in the series.